Hello, ladies and gentlemen. Thanks for tuning into our webcast today. I'm Cheryl Rogers, the Director of Marketing here at Golden Helix, and today Gabe Rudy, our VP of Product and Engineering, along with Nathan Fortier, a Senior Product Engineer, as well as a Field Application Scientist here at Golden Helix, are going to introduce you to our new copy number variant calling capabilities in VARSeq. Gentlemen, I'll hand it over to you.
All right, thank you, Cheryl. So I'm very excited today to be sharing with you the work we've been doing on creating a CNV calling algorithm that will be running in our VARSeq platform to call CNV events on targeted regions such as those in gene panels, etc. So, and before I get started too much, I also wanted to give you a little bit of a background about Golden Helix. We had a fantastic number of people register for this event, so some of you may have not been to a previous webinar, and so I just want to give you a little context there, but also motivate essentially why we are calling CNVs in this type of NGS data, why this particular type of NGS data, how it applies to the clinical workflow, etc. Once I do that, I'm going to pass this over to my co-presenter, Dr. Nathan Fortier, who is really the mind behind this algorithm, and he will present the high-level overview of how our method works, how it is unique, and how it builds on some of the ideas of existing CNV calling algorithms, and then does novel things that we're able to do inside the VARSeq platform that have not been done by other algorithms in the literature. And then, of course, showing is better than just telling, so we'll give a demonstration of our upcoming version of VARSeq that has this new method integrated, so you can really get a sense of how not just the algorithm works, but the interpretation process, now that you have both variants and CNVs in your VARSeq workflow, how you can use our fantastic visualization at Golden Helix, our reporting technology, etc., to really integrate that into your existing lab workflow. And then after that, I'll wrap it up. We'll talk a little bit about the availability of this method and some thoughts in going forward. So with that, let me give you, and finally, before I get started, there is a questions pane in your little GoToMeeting docket. We will spend some time at the end going through questions, and if we don't get to all of them, we'll be happy to follow up with you afterwards. So feel free to type those in as we go, as they sort of come up as you're watching here, and then we'll spend some time at the end going through those. So a little bit about Golden Helix. Golden Helix is a track record spanning all the way back to 1998 of serving not only the clinical market, but the research and pharma market as well. One of our early stakeholders has been GlaxoSmithKline, and at the moment this is essentially how the Golden Helix product portfolio looks. On the left we have VARSeq. This is our flagship clinical applications product for doing variant filtering and annotation, setting up your workflow to be able to interpret the variants, being able to place those into a report context, being able to define that workflow so that it can be rerun flawlessly with all of your lab-specific knowledge and know-how built into that workflow. And we have ways of automating that running in a pipeline mode. To the right of that, we have VS Warehouse, and this is a fantastic place to start to collect your samples over time. So as you're running your clinical workflows, you're collecting those samples. Those samples are a treasure trove of data, and Warehouse is a centralized place to store that data in a versioned manner where you can use them for annotations and also to store things like your reports that you're generating on your samples, and have that be a central place of sharing and integration. And finally, we have our SNP and Variation Suite, which is our research platform for doing large N analysis. So that covers things like association studies, mixed model analysis, genomic prediction, across agrogenomics, so plant and animal as well as human. And CNVs is not a new topic to us. We have a CNV calling algorithm in SNP and Variation Suite, but it's a very different beast. It's designed around microarrays, large numbers of samples, etc. And so what we're adding today to VARSeq is single sample CNV analysis. So being able to really interrogate a single sample and have a high precision CNV calls that make sense, that are designed around NGS data, not microarray data. So Golden Helix's success in being able to build these products is really a collaborative effort with our fantastic partners and customers across various industries, so reaching from, again, clinical to research to government. And with this essentially is the ability for us to continually adapt and build things like this new algorithm by having customers who are able to collaborate with. And so today we're going to be talking about this method, and the data that we'll be using later on, we'll mention this, was provided by our customer at Prevention Genetics. So this is real clinical data with validated results that we're able to build something like this that is going to be applied into a clinical setting. So not only do we have that track record and that portfolio of customers, but we've been cited in over 1,000 peer-reviewed publications, including some very prominent ones. So essentially we have this track record, and it builds off of our business model of essentially having a subscription model bundled with support, training, and that collaborative effort to improve the product over time. So that's what you're getting when you are looking at Golden Helix products. Okay, so that's our background and a little bit about the topic today. So we're going to be talking about calling CNVs on NGS data, and in particular, we're focusing on targeted gene panels. So these are your typical, maybe an oncogene panel, hereditary gene panel, maybe covering 50 genes, hundreds of genes, but these are the things that are actually being used in a clinical workflow today. This is both from an economics reimbursement perspective and from a testing pragmatism. We see gene panels being used. There are a lot of existing samples that people have started to collect over time, and we're looking at, is there just value we can add by calling CNVs on that existing data? So beyond just the value of having this extra data, NGS data provides a precision that you wouldn't see in your standard chromosomal microarrays or other types of assays, where we're able to make a call on an individual exon, which is generally the same as a target region, and make that call with very high confidence versus your sort of backbone probes across the genome. CMA, you're looking at maybe only making calls above 100,000 kilobases. We're talking on making calls on a single exon level, which may only be hundreds of bases wide. But there are challenges, and the calling algorithms we're talking about today, and essentially all NGS calling algorithms, are based on looking at the coverage of reads for these given regions. And as you can see, just in this little region here, it goes up, it goes down, it goes up. Are these CNV events? No, this is just the systematic differences that you get from target to target when you are doing a target capture. So there's differences in the target capture binding, there's differences in GC content, there's just PCR amplification. There are all these things that provide the systematic biases for one exon region versus another, and so you can't just look at this raw data and call CNVs. You need to have a method that is able to normalize this, to give you a proper baseline, and what we'll be talking about today is that method. And as long as you have reference samples, which in a clinical sample, in a clinical pipeline, all of your samples can be used in the reference context, simply because CNVs are so rare that the vast majority of the samples you see are going to be normal for any given target. You can start to do this type of work. And even if we do call some false positives, like any method will, especially at the target gene panel level, there's going to be so few of those that it's going to be very feasible to manually review those, just like you manually review the dozens of variants or a couple of variants that come out of the interpretation pipeline. So that gives you an example of, or the idea behind why we are building this type of method and what we expect it to be used for. And so with that, let's go ahead and pass this over to Nathan, who will give you the background on this method and demonstrate it in Varsik.
So CNV detection typically involves 4 key steps. The first is normalization. That is correcting for general noise in your coverage data. The main approaches in the literature are either PCA, that is principal component analysis, or correcting against match controls. And in our case, we've chosen to use match controls. Many algorithms do some sort of data correction that is correcting for systematic bias in the data, either in terms of GC content or low mappability regions. But once you've got your data normalized and corrected, the next step is actually to perform the classification. That is, you go through your target regions and you determine if they're diploid, homozygous deletion, heterozygous deletion, or duplications. And this is usually done using either simple thresholding approaches or using hidden Markov models. And once this classification has been performed, you need to perform what's called segmentation. And that is going across your targets and determining what regions are continuous CNV events, generally by merging targets or by segmenting regions based on coverage profiles. And there's a number of approaches that exist for each of these steps in the literature, but all of these approaches ultimately rely on coverage information to call CNVs. The two main approaches in the literature for CNV calling are thresholding. That is, you take one or more measures, usually they take multiple measures, and you combine these measures using logical operators. And if all of the measures cross a certain threshold, you'll call a CMV state based on that threshold. So for example, maybe you're using a z-score and a ratio, and if they all exceed a certain value, then maybe you call the CMV state as a duplication. Or maybe if they're below a certain value, you call it as a deletion. Hidden Markov models are different. Hidden Markov models are probabilistic models. Specifically, they're Markov processes in which you have a hidden state that you're interested in. In our case, this would be the actual CNV state of a particular target. And you have an observed state that is your evidence. In the literature, they often use a z-score for this. And you call CNVs by observing your evidence, by setting your evidence in the model, and then performing inference to determine the most probable state assignment for your hidden state variables. One big disadvantage to hidden Markov models is that it's very difficult to incorporate multiple metrics into these models. But it has the advantage of providing a probabilistic method for determining CNV state. Because of this limitation with hidden Markov models, the limitation that they can't easily incorporate multiple evidence metrics, it led us to use a different probabilistic model for our approach to CNV calling. Specifically, we used dynamic Bayesian networks. The idea behind a dynamic Bayesian network is that you have, it's a directed acyclic graph in which you have nodes that represent random variables, and you have edges that represent conditional dependencies between these variables. And each node in the network encodes a conditional probability distribution that's conditioned on the node's parents. And so here on the right, what you see is a network that's very similar to the dynamic Bayesian network that we use in our approach. We have a CNV state that is unobserved, that is, it's hidden, that we're trying to determine the correct state assignment to. And we have three evidence variables. The first is a z-score relative to the controls, and that is just measuring the number of standard deviations away from the mean normalized coverage for each of the controls. We use a ratio, that is just the ratio of the normalized coverage for the sample of interest divided by the mean normalized coverage for our controls. And then finally, we incorporate B allele frequency as evidence in our network as well. B allele frequency is particularly useful because it can provide us both evidence for duplications as well as evidence against deletions. And Varseek already computes this information, and so we already have it readily available to us. For duplications, you expect to see certain variant allele frequencies for variants within a given region, if that region is in fact a duplication. And you'd expect ratios like 1/3, 2/3, etc. And you can see an example of this in the picture on the right here, where we have a duplication. There's a variant within this duplication with a B allele frequency of almost exactly 1/3. And this provides our Bayesian network with evidence that this duplication is in fact a real legitimate duplication. But additionally, we can also find evidence against deletions, because any value that's not 0 or 1 is evidence that what we're looking at is not really a deletion, even if the z-score or the ratio may indicate that it is. And this can be seen in this next figure, where we have a variant, what would normally be called as a copy number variation in this region. It looks like it might be a deletion. But there's a variant in this region with an allele frequency very close to 0.5. And that kind of value is basically impossible if this is a true deletion event. And so that provides us pretty strong evidence that this is really not a deletion. And it would provide our Bayesian network evidence to not call this as a deletion and avoid this false positive. So next, I'm just going to give a brief overview of the various steps involved in our algorithm and how it works in the context of varseq. So in varseq, you'd begin by importing your variants, importing your BAM files, and then you would compute coverage over your samples. And this coverage information is what's going to be used as input into the CNV calling algorithm. Next, when you actually run the CNV caller, you have the choice to add the current samples in your project to the reference set. So in varseq, what we do for you is we maintain a library of reference samples. And these reference samples are used during the normalization procedure in order to correct for noise in the data. And so every time you run samples through your CNV caller, you can actually add these samples to your reference set and grow your library of reference samples that will then be available the next time you run the algorithm. Once you actually run the algorithm, the first thing it does is it picks 30 match controls. These are the 30 reference samples that are the most similar to the sample of interest. It then computes the ratio, z-score, and B allele frequency information across the targets for the sample. It classifies the targets using our dynamic Bayesian network. And then finally, it performs event segmentation, where it merges adjacent targets in order to obtain continuous CNV events. And then this information is outputted to a table. And so what you end up with is a target region table that has all of your target regions with the coverage information along with all of the CNV information. So whether the target was called as diploid or deletion or duplication, as well as information about the various metrics we use, such as the ratio and the z-score. You also obtain an event table that actually lists the individual copy number variations that were called, along with information associated with those. And we provide you with additional information in your sample table, providing summary CNV information associated with each one of your samples, things such as how many CNVs were called for this sample, etc. We've evaluated our algorithm using a set of benchmarking data provided to us by Prevention Genetics. This data includes 144 cancer panel samples. And these samples cover 549 target regions and 31 different cancer genes. Of these regions, 91 fall within CNV events, including heterozygous deletions as well as duplications. We've run the algorithm over 48 of these samples using all 144 samples as controls. And what we're presenting here is some experiments that illustrate the effect of the CNV probability, that is, the prior probability of a copy number variation prior to observing any evidence. These experiments evaluate the effect of that on sensitivity, that is your true positive rate, and precision, that is the positive predictive value, which is related to the false positive rate. So the first set of results that I'm going to show you are shown here in this table. And what we have here is the prior probability of a CNV event. And this parameter is tunable through varseq. And so you can tune this to whatever value you feel is right for your lab. And what this table illustrates is the effect of this prior probability on the sensitivity and precision. If you have a very low prior probability of a CNV, you're going to get fewer true positives, and as a result, you're going to have lower sensitivity than you would have if this value was higher. However, you'll have higher precision and fewer false positives. As we increase the prior probability of a CNV, the sensitivity will increase, that is, you'll call more true positives. At the highest end, with a value of 10 to the negative 6 for our prior CNV probability, we actually correctly call 100% of the CNVs in this data set. However, this does have an effect on precision. That is, as you increase the prior probability of a CNV, your precision will go down and your number of false positives will go up. So in order to mitigate some of these false positives, we've added the ability to flag low-quality events through varseq. And you can actually filter out these low-quality events if you would like, and only look at high-quality events, thus reducing the false positive rate pretty drastically, as you'll see in a moment. The metrics we use to determine if an event's low quality include the depth of the controls. That is, if all of your control samples have very low mean depth at a given region, then any calls in that region are likely to be unreliable, and we flag that region. We also use the variation between the controls. That is, if the coverage at a region varies wildly between the controls, then calls at that region are likely to be unreliable, and we flag that region as high control variation. And the final flag that we provide is within regional IQR. And this flag just indicates that an event at a given region can't really be differentiated from the noise at that local region, and therefore is likely to be unreliable. An example of this is shown in the figure below, where we have called a het deletion, but this region is particularly noisy. And you'll see, especially if you look at the z-score, there's a number of exons in this region that fall in this really low range, but are probably not deletions. And likewise, we have a lot of events that have unusually high z-score. So because this region is so noisy, and because this event doesn't really fall significantly below the noise, we flagged it as being within the regional IQR. So if you apply these flags and you filter out all the events that are flagged, you drastically reduce the number of false positives and drastically improve the precision of the algorithm. And this is illustrated by this table. So we'll see here that when the prior probability is 10 to the negative 12th, our precision is now 98.8%. We're only getting one single false positive. We've eliminated 30 of the false positives that we had obtained previously prior to flagging these CNV events. And this trend continues throughout all of the various values for the prior probability, all the way up to 10 to the negative 6, where we had eliminated 54 false positives and are only obtaining 9 false positives. Now, it is possible that some true CMV events may be flagged. And in fact, when the value is set to 10 to the negative 6, there are 3 CNV events that are true CNV events that were flagged. In this case, they were flagged because those regions where the events existed had very low coverage. And so, it may make sense to look at some of these events if you know a region just happens to be noisy. It may be worth it to investigate further, even if an event was flagged. But by and large, it drastically improves the precision of the algorithm. So with that, I'm going to go ahead and jump into VarSeq and show you what the algorithm looks like in practice. So what I currently have on my screen is the initial VarSeq screen. This is what you're going to see when you first launch the application. And what we're going to do is we're just going to create a new project. This is going to be an empty project. Generally, what you would do is you would actually use an existing template that you had created that applies filters and annotations and that sort of thing. But for now, we're just going to import variants into an empty project with no filters or annotations. So we'll provide a name for our project. And the first thing you're going to want to do when you create a VarSeq project is import variants into the project. So we'll select the variants for our samples. The next thing that you need to do prior to importing your variants is associating a BAM file. Because the CMV algorithm uses coverage information, we need to pull that coverage information from the BAM file. And so if the BAMs already exist in your folder, as they do here, the BAMs will automatically be associated with your VCF files. And so they're associated here. I'm just going to click OK. And now we've associated our BAM files. Here, as you can see on the right, I click Next, click Finished. And now VarSeq has imported all of our variants. And so we can go ahead and hide our filter chain. And we can go ahead and compute coverage over our samples. So we'll select Target Region Coverage. Hit OK. And now we'll select a track, and this just defines the regions over which to compute the coverage. And so I have a cancer gene panel file here that was generated from a bed file, and so I will use that. Click OK. And now the algorithm has begun computing coverage over our samples. I should also note that here in VarSeq, you can also, as well as looking at variants, you can also look at information at the sample level. And you can provide any information you want to related to specific samples, and we can import that information and make that available to you here in VarSeq. And when coverage actually finishes computing, we provide summary coverage information associated with each of the samples. So for example, the sample mean depths, as shown here, are provided, and the percentage of regions covered at various coverage levels is also provided. So now that we've got our coverage computed, we can go ahead and call CNVs over this data. So I'm just going to click Add Computed Data. I'll go ahead and select our CNV Calling algorithm and click OK. And here we have a dialog that allows us to configure and customize the algorithm. Basically, every parameter associated with the algorithm, including all of the distributions associated with the dynamic Bayesian network, can be tuned. But for now, we're just going to worry about the probability of a CNV. And for most applications, this is the main parameter that you will tune. At a very low value, such as 10 to the negative 12th, you'll have high precision but lower sensitivity. And at a value like 10 to the negative 6, you'll have lower precision but higher sensitivity. That is, you'll get more true positives, but you'll also get more false positives. So for now, we'll just leave it at the default value of 10 to the negative 12th. We've selected to add these samples to our reference set so they can be used as controls. So we'll just click OK and run our CMV algorithm. So it's finished now, and it has called a number of CMVs for some of our samples here. So let's go ahead and look at some of these CNVs that have been called. So here we have sample number 2, and we'll see that a heterozygous deletion has been called for sample number 2. Well, we can go ahead and actually plot information about these CNVs and look at them in Genome Browse to get a better view of if these are in fact real CNV calls and what they look like in context. So first we'll go ahead and plot our coverage information. We can go ahead and hide this. And the next thing that we're going to do is we can go ahead and plot our CNVs. So we can right-click here and plot the CNV state for our current sample. And we'll see all of our CNV calls showing up. We can also filter down to look at only CNVs that actually have a CNV state assigned. We can do that by clicking Query Column Values. And we only want to look at CNVs that were either called as a deletion or as a duplication. So now we can see we have a single CNV call here of a heterozygous deletion. And we can also plot information from our coverage table. So for example, we could go ahead and we could plot the z-scores. And we could plot our ratios so that we could get an idea about exactly why these were called. Okay, so let's go ahead and look at this heterozygous deletion here in this sample. We can go ahead and we can actually sort by CNV state, and we can see this heterozygous deletion that was called here. And if we zoom out, we can see the context. And you can see that in this region we have a drastic drop, both in terms of ratio, the ratio being at about 0.5, which is exactly what we would expect for a heterozygous deletion. And we have a z-score that is very low, all of which are over 3 standard deviations away from the mean of the coverage samples. And so this looks like a real heterozygous deletion. So let's go ahead and look at another one of our samples. So let's look at sample number 22. So you'll notice that as we switch to sample 22, our plots automatically update to reflect that sample. And we'll see that for sample number 22, we had 2 CNVs that were called. The first is a duplication event. Let's look at this duplication event in sample number 22. So we'll see it's a single exon event. And in fact, if we zoom out, we can see the context here. And the thing to notice is that, especially in terms of z-score, this event does not appear to be differentiating from the noise in this region. In fact, there's a number of events that have in this region that have very low z-scores and very high z-scores. And so there appears to be not only pretty high variation, and of course we flagged it as having high variation among the controls, but it also doesn't appear to exceed the regional interquartile range requirements. And so we flagged this variant as being likely unreliable. So the next thing I would like to do is go ahead and look at a project that looks a little more like a project that you would use in a clinical setting. Generally what we would have is we would have filters applied, we would select variants to include in reports, and that sort of thing. And we'd apply annotations. And so let's look at a project that looks a little more like a typical clinical workflow. So in this project, the first thing you'll notice is that on the left we have a filter chain, and what we've done is we've filtered down to a set of interesting variants that we'd like to look at. We also have a report form opened, and we've added some variants to a report. Here on the right, you'll notice these two checkboxes, and these checkboxes are used to add variants to a report or remove variants from a report. And so here, you'll notice that as I unselect this checkbox, a variant has been removed from our report here on the left. And if I select it again, our variant will be added back to the report. Well, we can do a similar sort of thing with copy number variations. We can actually include copy number variations in our reports. And so, if we move over here to our copy number events, we'll see that in this sample, sample 13, we have actually called 2 copy number variations. So let's look at these copy number variations and see if they appear to be legitimate. Well, this first one was flagged as having high variation between the controls. And so let's go ahead and inspect this variant and see if it looks like a real variation. So we'll see it spans 2 exons. But as we zoom out, we can see the context, and we do see a bump here in terms of the ratio and in terms of the z-score, but the z-scores are fairly low, and they're not really differentiating from the normal z-score of 0 by a large amount. And this is to be expected, because if there's high variation between the controls, as the flag indicates, then we would expect there would be high standard deviation amongst the controls at both of these positions. And that would in turn dampen the z-score. And so it's pretty likely that this duplication is not a real CNV event. Next, we can go ahead and look at this other duplication in chromosome 13. This is a duplication in BRCA2. It spans 4 targets, that is, it spans 4 exons. And you'll notice that both the ratio and the z-score are quite high. The z-score indicates that all of these are between 2.5 and 3 standard deviations from the normal compared to the controls. And the ratios are quite high. They're basically exactly what we would expect to see in terms of ratios for a duplication. So this is probably a real event. So let's go ahead and include it in our report. So we can select the event, and you'll notice that it has been added over here to the left and been included in the report. And for any variants or copy number variations in your report, you can change anything in the report that you would like. So for example, maybe we want to set this to likely pathogenic because it's not a copy number variation that we know to cause any specific disease. And of course we can edit the interpretation if we would like as well. And once you're happy with the report and you've included the all the variants and CNVs that you would like, we can go ahead and click this button here, generate the report. And you'll see that a report's been generated. It includes 4 different genes, including BRCA2, the gene in which we had our copy number variation. And if we scroll down here, we'll see that our copy number variation has in fact been included in the report. We have a 4-exon duplication spanning around 9,000 base pairs in BRCA2 that is likely pathogenic. And so that gives you an idea of how you can use the CNV calling algorithm in VARSeq and incorporate it into your clinical workflows. And with that, I would like to go ahead and pass it back to Gabe Rudy, who can tell you a little bit more.
All right, thanks, Nathan. Fantastic overview. So hopefully you guys, just by watching this and seeing a little bit of the high-level description of the algorithm and how it works in VARSeq, get a sense of what the application is that we're trying to target here. We think that there are unique things about our algorithm, mostly in the ability to include multiple metrics. So we're not just looking at a z-score, but we're looking at the z-score plus a ratio, plus any overlapping variants if there are any. And so that is something quite new and novel, and we find that all those extra metrics do essentially help with this tunable sensitivity precision trade-off. And so that's what we are evaluating in the context of these gene panels. We are in an early access mode. We have a couple clinical partners that are looking at this, evaluating it with their own sets of data, essentially with their own historical archive of samples, with their own known CNVs. And just like any clinical workflow, we expect that you should be looking at this in the same way, validating it against any known CNVs, or calling CNVs and using some sort of orthogonal assay to validate their validity. We think that this robustness of being able to include multiple metrics does scale and does work outside of just the gene panel that we presented here today. But of course, like anything, it needs to be battle tested, etc. So when we are done with that early access, we're going to be shipping this in our next version of VARSeq 142. And it's going to be part of our VARSeq clinical package. There is already a question in the Q section about exomes, so I might as well answer that now. We think that this strategy does scale up to exomes. Fundamentally, it's the same idea. We are making calls at the single target event, but those if you have a whole chromosome duplication, you will be able to expand that call across and get to that size of event as well. So scaling from single target events to multi-exon events to multi-gene and chromosome level events. But the caveat here is we know that there is a lot of regional effects in exomes, and we expect that we need to treat this just like we have in this pass, work with our clinical partners, benchmark some data against known samples, and work on making sure we have proper QC flags, potentially regional knowledge. So how do we treat things like highly copy number variant regions of the exome that have known high copy number counts in control samples, MHC region, etc. So that's something we'll be looking into. Certainly, if you are interested, you can continue. You can treat it like an investigative method and try it on your exomes as well. And with that, we do have a good chunk of time, and so we'll save some here for going over some of the questions that have been trickling in through the webinar. I've flagged a couple already, so Cheryl, I'll pass it over to you to moderate the questions.
Thanks, Gabe. Before I get to those questions, I do have just a couple of quick announcements. As always, I will email out the link to the webcast recording. You should see those on Friday. Our next webcast will be on Wednesday, November 9th, and invites for that will go out in just a few weeks, so keep your eyes on your inbox. Again, if you're attending ASHG this year, we would love to meet you, see you. Come by. We're in booth 902. If you've attended ASHG before or any other event with us, you know we give away some pretty awesome t-shirts after our in-booth demos. I will be posting a blog tomorrow that has that schedule, so you'll know when you can receive a t-shirt. So that's it for me. Let's get to those questions. First question here, is the CNV a standard feature of VARSeq?
So, right, as I mentioned, this is going to be part of our clinical package. We really think this makes sense alongside having the ability to design your own custom reports. The one we gave you today was just an example. You can design reports that are customized to your labs, both in terms of presentation and in terms of content. We have other webinars in which we go through sort of our clinical report and pipeline, our clinical package of VARSeq, so please check those out if you're interested. If you are interested in this package, we have actually a promotion that's going on right now. So we have a 2-seat VARSeq plus reports plus the CNV Calling package that would be relevant to you, and it comes in at just under $15,000 at $14,995. And often what we do is, this is a yearly subscription, but given this is a new method, we want you to get up and going with low risk. So this is actually a 15-month package at that price, so you don't even have an extra renewal next year. So keep that in mind and reach out to us if you're interested.
We are interested to join the development as a beta client. What is the process to participate? Excuse me.
Right, so as I mentioned, we already have some clinical labs that are validating this in that early access mode. So we have received requests like this. What we're going to do is just limit that to existing customers. So and existing customers who have this report package. So if you're interested, if you are an existing customer with this report package, please contact us. If you're interested in getting that package, contact your area director at Golden Helix.
What metrics are used to determine which 30 samples are most similar to the sample of interest?
All right, I'll let Nathan answer that one.
So we borrowed our approach for selecting match samples from an algorithm called Confiding. You can find it in the literature. The basic idea here is that we want to find samples that have a similar coverage profile. So what we look for is samples that have similar normalized coverage across the target regions. And basically we come up with a score based on the normalized coverage across the target regions for the control samples. For each one of the controls, we then sort them by this match score that's based on normalized coverage. And then we select the 30 at the top of this sorted list.
All right, sounds good.
Thank you. Can this algorithm be used equally well on germline and somatic samples, or is this germline only?
That's a good question, and I'm going to bundle in with that a couple other questions that were about tumor-specific content. So fundamentally, these metrics are very tunable, and so there may be a different expected profile in a tumor-based gene panel where you might have extremely high copies of a number of CNVs, and so we can say adjust your expectation on the what a call for duplication is, but fundamentally this method should apply with the caveat you might need to tune it to match your historical samples and what you see there. Similarly, the things like the B allele frequency, if you expect to have sort of somatic mutations, of course those range in their frequency, and if those are being called by your CNVs, we'll be able to have a parameter to say not include B allele frequencies in the calculation. So some of these things are intended to be parameterized to make it specific to your workflow. And like everything in VARSeq, once you define and tune that, you can save out this project as a project template. The next time you run it, all those parameters, including all those beautiful plots, and you saw the last one Nathan went to, we had the prior thresholds as lines on those plots, and we had steps connecting the dots, things that really just help in the interpretation. All that is saved in your template, so every time you create a new project with a new sample using that template, you'll get all of those algorithm parameters and interpretation parameters.
Although you have a reference set, since the sequence results will depend on the methods and platform, do we have to run the reference samples in your set in our own platform to get the sequencing data? Will different targeted panels have different sets of reference samples?
Yes, and so that's a very good point. So we can't provide you with pre-baked reference samples. Even if we wanted to, you really do need to have those that are being run using the exact same target capture kit and preferably the same sequencer. We have tested this across multiple runs, and we think the run level effect is handled by the normalization, but certainly as soon as you do things like change the target capture and you add an exon or delete an exon or add genes or delete genes or are not comparing the same set of targets, all bets are off in terms of being able to do this comparative-based approach. We automatically inform our reference samples with which target capture kit they were defined with. So you can certainly use this algorithm. On day one, you might be looking at one panel. Day two, you're looking at another panel. We will intelligently only pick reference samples that are matching the coverage profile of the project you're currently doing your analysis on. So we do handle that, but you need to be doing this in a way that's matched to a specific assay and a specific workflow on your lab level.
Is there a minimum sequencing depth recommended for running this algorithm? For example, would this work well for depth 50 to 100x exome sequence?
Right, and that's also a very good question, and ultimately we're going to ask you to validate it under any circumstance. So you saw some of the samples here, on average, were being called 2, 3, 400x. Some of them were being only at an average depth of about 60. When we start to get below that, we saw the false positives increase, and in fact, we provide not only QC flags at the individual call level saying we think this is a sketchy region, we also provide those flags at a sample level saying we think this sample is not behaving as expected. It either has very low read depth, high level variation, etc. So if you're starting to get into this 30x range, that may be a problem. But if you're in the 100x range, as most gene panels are, you should be good to go. Of course, validate based on your own protocols.
What is the minimal size of deletion duplication that's likely to be detected?
Right, that's a good question. Again, we were able to call with high precision or with high confidence single target events, especially ones that had metrics across these 3 different metrics all supporting that call. So that's actually very amazing, and because essentially a single target event has a very low prior at all of it existing. When we merge events, targets, when we start to see 2 or 3 exon events, they reinforce their probability with our second pass. So those are often called by other algorithms, but being able to call a single exon deletion or duplication is something we're pretty proud of, that we're able to have that sensitivity without dramatically increasing the false positives, as you saw with our QC filters, etc.
Thank you, Gabe. I think we're going to go ahead and wrap it for today. We've got a lot of questions. Fear not, we will get back to you with answers to all of those questions. You'll be hearing from us soon. Thanks, Gabe. Thanks, Nathan, for a great presentation. Thanks, everyone, for joining us. And I hope everyone has an outstanding day.